Hi friends, welcome back to my YouTube channel. In this video, I'm going to discuss and explain you about the DNA and RNA differences. So this answer comes as a long answer. Sometimes it comes as a short answer. So here I'm going to give you the 20 differences of DNA and RNA, which are as a long answer. So if you study for the long answer, it is also helpful for you as in a short answer. So let's start this video without wasting time. And before starting this video, let me tell you, if you are new on my channel, please subscribe my channel and share my channel with your friends. Let's start a video. So first different will be going to start with the DNA. That is, DNA, we know that it is a genetic material, right? So DNA is a genetic material in all living organism, right? DNA is a genetic material for all living organisms. And if we talk about the RNA, RNA is a genetic material for viruses. So RNA is a genetic material in some viruses. And if we talk the main differences between the DNA and RNA is, what is the main difference? Can you say? With the name itself, we can understand it has a difference in sugar, right? It has a much different in a sugar. So what is the difference? It is a deoxyribose sugar. So DNA has what? Deoxyribose sugar, while RNA has ribose sugar, right? Now we'll be going to see the structure of DNA and RNA, like which shows the difference between the deoxyribose sugar and ribose sugar. Here we'll be going to draw the structure of RNA and DNA. Here the same structure will be there, only one difference will be there, oxygen difference will be there, right? Only oxygen difference will be there, that's it, nothing more. So here you can see in the in the DNA there will be a H here, only H here, OH, H, H. If you know the structure, you can directly draw the structure, right? Here H and here CH2 OH, and this will be your H. So this is the first carbon. Let me draw it with another pen. This is the first carbon, this is the second carbon, this is the third carbon, this is the fourth, and this is the fifth carbon. At the second carbon, it has a deoxyribose sugar. This we call it as ribose, right? And this one is deoxy because oxygen is not present. And if we see in the ribose structure, what is the difference is it has OH at the second carbon. Same structure will be there, only difference between the oxygen occurs, CH2OH and here H, right? So here you can see at the second position it has a OH. It is not a deoxy. Why we are calling deoxy? D means removal of oxygen in the ribose sugar. That's why it is deoxyribose sugar. Now we'll be going to talk about the alkaline condition. So what DNA is? DNA is stable under alkaline condition, while RNA is not stable. Why? What is the difference? RNA is not stable under alkaline condition. Now tell me, what is the difference that makes the DNA stable while it makes the RNA is unstable for alkaline condition? That is only the difference. It is deoxy while it is in ribose. It has no oxygen. So what happens when phosphate group attacks? Okay, phosphate group attacks to this attacks to this part of oxygen and hydrogen. This will form a phosphate group, but from here oxygen and hydrogen reacts with this phosphate and forms another bond with this phosphate and makes it not stable for the condition, whereas it is not having oxygen. That's why phosphate group easily attacks on the on this oxygen and hydrogen and easily forms a group, but it will be a stable form. Here it is not becoming stable, it is becoming unstable due to this oxygen present in the RNA. Okay, now we'll be going to see the fourth difference between the DNA and RNA. That is, DNA is a double stranded, right? DNA is a double stranded except in few viruses. Am I right? In few viruses it is a single stranded, but in all the living organism it is a double stranded. And if we talk about the RNA, RNA is a mostly single stranded. RNA is a single stranded except in some viruses. In some viruses it acts as a double stranded, so we are accepting that, and in all the forms it is a single stranded. Now we'll be going to talk about the length of DNA and length of RNA. If we talk about the length of DNA length is quite large, right? Length of DNA is a quite large, which consists of millions of nucleotides. Yes, obviously it is true, it consists of million of nucleotides, whereas if we talk about the length of RNA, it will be very short. Length of RNA is short, right? It consists of thousands of consisting of, we can say, few thousands of nucleotides. This is 100% true, because DNA is present in all over the living organism, and that's why length is millions of nucleotide. Length consists of million of nucleotides. And if you don't know, nucleotide is consist of three things, that is ribose sugar and nitrogen base, and also it consists with a phosphate. And if we talk about the nitrogen base and ribose sugar, these two constitute as a nucleosides, right? Now we'll be going to talk about the sixth main difference between the DNA and RNA is bases. We talk about the bases now. So here bases are adenine, thymine, guanine and cytosine. And if we talk a basis here in RNA, that will be a adenine. In place of thymine, here uracil comes, and guanine, cytosine, right? So now we'll be going to talk about the seventh difference, that is purine and pyrimidine ratio. Here purine and pyrimidines are equal in ratio. That will be 1:1 kind of ratio. And here purines and pyrimidines are not equal. Why? Purines and pyrimidines are not equal. Why? What is the reason why they are not equal? Why? Because in most of the cases RNA is a single stranded RNA. In most of the cases, not most of the cases, in all, RNA is a single stranded RNA, whereas DNA is a double stranded. That's why they are having the basis and complementary basis, that to complementary basis of purines and pyrimidines. Complementary basis of purines and pyrimidines, due to that DNA is having a equal ratio of purines and pyrimidines, while it does not have equal ratio because it has a single stranded of RNA. When it is having a single stranded of RNA, when purines and pyrimidines will be equal, when the strand is complementary, see, when the strand is single, how the purines and pyrimidines will bind? When the strand is complementary, both the bases will be in equal ratio, right? That's why it has a not equal ratio and not equal pyrimidines and purines. Okay, now we'll be going to talk the eighth difference between this. Here adenine binds with thymine, and here Adenine binds with uracil. Isn't it that true? Yeah. Now we'll be going to see the another difference, that is DNA occurs in how many forms? Here DNA occurs only in one form in a organisms. Okay, DNA occur in only one form in organisms, while RNA occurs in many forms. So what are those many forms we'll be going to see? Here RNA occurs in three types. Those are mRNA, tRNA, rRNA. Okay, this is short RNA. Okay, small r. Okay, RNA. Now we'll be talk about the 10th difference of the DNA. Okay, here DNA occurs in nucleus, right? Where the DNA occurs? DNA occurs in nucleus, nucleolus, extrachromosomal DNA, that too in mitochondria and chloroplast. Mitochondria and chloroplast. If we talk about the RNA, where it will be present? If we talk about RNA occurs in nucleus, nucleolus and cytoplasm. That too in nucleolus, mRNA will be present. Okay, in cytoplasm, tRNA and rRNA will be present, right? So these are the 10 differences in the DNA and RNA. Here you can see these are the 6 differences, and here I showed the 10 differences. Now we'll be going to see the 11th difference between the DNA and RNA. 11th one will be DNA present in chromatin, right? Here the RNA present in ribosomes. Isn't that true? We have studied many times that RNA is present in ribosomes. RNA synthesize the ribosomes. We have studied it many more time in the 12th standard, right? Now let's see another difference, that is DNA is self-replicating. Now we'll be going to understand this concept behind self-replicating, how it is called as self-replicating. While RNA is synthesized from DNA when needed. Okay, when needed, it's synthesized from DNA. So we'll be going to understand the concept behind this self-replicating. Here you have see the central dogma of life, right? So what we have a DNA, and DNA will gives RNA, and RNA gives a protein, right? DNA on transcription, it will give you the RNA. On reverse transcription, RNA will gives you a DNA, right? And on translation, RNA will give you a protein. So this is a central dogma of life. And here we have drawn some symbol like this, right? So it shows a replication. So DNA on replicating by itself, it will give the DNA gene. Okay, it will produce a DNA again. But on transcription, they will produce a RNA. So if RNA needed, DNA has to synthesize the RNA. Okay, RNA can synthesize the DNA when first DNA synthesize the RNA, then only RNA can resynthesize back to the DNA. Did you get that? on reverse transcription. First, DNA has to initiate the cycle. That's why DNA is called as self-replicating and RNA is synthesized from the DNA when needed. Right? So this whole process, we called it as central dogma of life. Am I right? Now we'll be going to see the difference between one more difference, which is very basic. We can write that also if we want. Like thymine is present in DNA. Thymine is present in DNA. And if we talk about RNA, we can write the uracil is present and thymine is absent. Right? Here you can write as uracil is absent. Okay, this is also the difference. So there are total 13 differences I showed. Now I'm going to show you more till 20. Let's see the 14th difference about the DNA. Here DNA on UV radiation. Okay, on UV radiation, it is susceptible. Okay, it is susceptible to UV damage. Whereas RNA, if you talk about the RNA, it is resistant. RNA is resistant to UV damage. Why it is susceptible and why it is resistance? Because DNA is a deoxyribose sugar and it is easily broken or harmed by the UV radiation, whereas RNA is resistant, unbroken or unharmed by the UV radiation. Okay, by the UV radiation. Now we'll be going to see one more difference, that is complementary nucleotide. If we want to talk about the complementary nucleotides, that is purines and pyrimidines, here in RNA it is present throughout the length of the DNA. Right? Am I right? If we draw this structure, okay, this is the DNA. Here I am drawing very badly. So here to the whole length there will be a complementary base pairs to each other, and that nucleotide base pairs are present throughout the length in DNA. While in the RNA it is not like that. It will present in the regions, some regions which is having a complementary nucleotides. Okay, nucleotides pairs, and sometimes it forms a hairpin like structure, at which place tRNA. If we see the tRNA structure at that time, we'll be going to see at the initial point of the tRNA, they will form a hairpin like structure, right? Hairpin like structure. So when we are going to see the types of RNA, we'll be going to discuss that hairpin structure in the tRNA. So this is also a difference. Now we'll be going to see the hydrogen bonds. Hydrogen bonds are formed between complementary bases of the opposite strands. Obviously it is true, when these strands are forming, there will be some purine and then pyrimidine, right? Between this there will be a hydrogen bonds which are bonding. So this is a 3 hydrogen bonds for guanine and cytosine, and there will be a 2 hydrogen bonds for adenine and thymine, right? So this is for a guanine base pairs with 3 hydrogen bonds, adenine base pairs with 2 hydrogen bonds. So there will be a hydrogen bonds formed between the complementary bases of opposite strands. So one strand is having adenine, other will have a thymine. So one strand is having guanine, other will have a cytosine with the opposite strand, with the opposite base pairs. Okay, do you understand that? And in the RNA, base pairing will be through hydrogen bonds occur as a coiled parts. If we study the tRNA, there will be going to, this is a tRNA structure, right? There will be a tRNA structure like this. So in the tRNA structure, there will be a short arm like this, right? So in that part, we can see how they are getting a coil. Here the amino acids will be present, and based on the base pair, that will going to add, right? So we'll be going to study this when we are going to study the tRNA. Okay, it will occur as a coil part. Now let's see the one more difference of DNA, that is helix geometry of DNA. So here helix geometry of DNA is B form. Okay, mostly it will be in the B form, and some are having A and Z also, A or Z form also there. A and Z form is also possible, but mostly it will be in a B form. Okay, and if we talk about the RNA helix geometry, RNA helix geometry will be in A form. It will be in a A form. Okay, now we'll be going to see the quantity. Okay, we'll be going to see the quantity here is fixed for cells. Now how I am saying that quantity is fixed for the cell or definite for the cell, because here as we are having base pairs equal, right, complementary to each other, and they are having 3 hydrogen bond means 3 hydrogen bond between these two, and 2 hydrogen bonds means 2 hydrogen bonds between these two, and they are complementary strands will be there, that is fixed, right? But here at some point it is a single stranded, and at some point it is double stranded, so there is no quantity is fixed. So quantity here is variable. We can say here the quantity will be a variable. Okay, now we can see, as we have saw the central dogma of life, I have to write this point there itself. As we have saw central dogma of life, we can write one more point here as DNA controls genetics and metabolism, including variations. Am I true? Yes, DNA controls the genetics as well as metabolism and variations by itself, because DNA is the only one which starts the cycle of central dogma of life, right? Like this we'll be having. Am I right? It is only controlling the process of RNA and protein releasing. It is only controlling the process of giving the DNA again. So it is responsible for genetics, it is responsible for metabolism, as well as for getting a variation in the protein. Am I right? Can we write the difference? And if we talk about the RNA,